Hi, my name is Jenny, and I'm a master's student at McGill University studying under the supervision of Dr. Valerio Hoyos Viegas. And today I'll be presenting my thesis project, the simulation of plant breeding strategies in the common bean, Phaseolus vulgaris L. So dry beans are a great source of proteins and fibers. They're low in fat and contain many vitamins and minerals. Some studies also show that they reduce the risk of heart disease and diabetes. As well, they improve soil health through nitrogen fixation. Dry beans are very important for many developing countries as they provide a source of protein and essential nutrients. While in developed countries, dry beans are mainly grown for livestock feed and export. Although in recent years, they have become more popular due to their nutritional value. So dry bean production in Canada takes place in Alberta, Saskatchewan, Manitoba, Ontario, and Quebec. And much of the dry beans produced are exported. Canada currently exports to 130 different countries. Rising global populations and the need for sustainable food sources has created an incentive to increase dry bean yields. So yields have been increasing linearly since 1956 and haven't yet reached a plateau. These increases in yield can be attributed to the introduction of new cultivars with improved traits. So how can we get these new cultivars? Well, we can do that with plant breeding. So plant breeding takes place over a number of years and can be quite resource and labor intensive. Many of the decisions that go into a plant breeding program can greatly influence its success. Computer simulations offer a way to obtain information that would not be possible empirically and can help plant breeders make decisions. As well, computer simulations let us look into the long-term effects of breeding programs. So the objectives of this study were to first compare 5 plant breeding strategies in terms of genetic gain, allele fixation rate, and haming distance in 3 selected traits. Secondly, to compare the average genetic gain per cycle in conventional breeding, speed breeding, and genomic selection. And thirdly, to investigate the genetic variance following conventional breeding or genomic selection. So here are some of the simulation platform and tools that I use. So the first is QGene, which consists of an engine and a module. I also use SimuPop, which can be used to simulate populations with a certain amount of linkage disequilibrium. R is used for data analysis, and Compute Canada I use for running the simulations as they can be quite computationally demanding. Here is the overall simulation pipeline. Starting with conventional breeding, I first have a QUG file that contains information on the genetics and environments of the population I plan to simulate. This file then goes into the engine, which produces some output files that then get run through the QLine Plus module to produce a number of output files, which are then used for data analysis. Speed breeding is done post-run, and genomic selection does contain a number of additional steps beforehand, and this involves training the population. So here are the simulation parameters. I use the linkage map reported by Galliano et al. Containing 1009 markers. Here's some other parameters. And I simulate 3 traits: seed yield, white mold resistance, and days to flowering, each with different heritabilities and number of QTLs. So before I continue, I thought I'd talk a little bit about an important concept in quantitative genetics, and that is the breeder's equation, where the response to selection or genetic gain is influenced by the heritability of the trait and the selection differential. The selection differential is then influenced by the selection intensity as well as the phenotypic standard deviation of the parental population. So looking at this diagram, we can see that this selection differential is actually the difference between the mean of the selected individuals and the mean of the parents. And then the response to selection is the difference between the mean of the offspring and the mean of the parents following selection. Here's another way to write the equation for genetic gain. In the actual simulation, we have the values for the mean offspring and mean parents, so we can actually use that to calculate genetic gain directly. So now I'm going to talk a little bit about the actual strategy, starting with the most simple mass selection. So F2 plants are grown and seeds are harvested in bulk. These seeds are then planted and once again harvested in bulk. And this is repeated until we get to F5 where we actually make some selection. And from there we go into the yield trials, preliminary and advanced yield trials. Just as a note, this is considered one cycle of selection. So next we have bulk breeding, which is similar to mass selection. The difference is that we keep the families separate. In single seed descent, rather than harvesting seeds in bulk, we take one seed per plant and use that to advance the next generation. For the pedigree method, we have early generation selection starting at F2, and you can see that we're already selecting individual plants. We're growing rows, selecting the best rows, the best plants within rows. And for the modified pedigree method, this is a combination of the pedigree method and mass selection. Genomic selection consists of taking a population that is both genotyped and phenotyped and training a model based on that population, then applying it to a testing population where individuals are only genotyped. These individuals are then assigned a genomic estimated breeding value, and phenotyping is not needed. Speed breeding is a way to increase the rate of development by lengthening photoperiods, and we can see that by using speed breeding, we can cut down the number of years from 8 to 5. On to the results. So this figure shows the genetic gain over 10 cycles. The vertical lines represent the point at which 90% of cumulative genetic gain has been achieved. So based on these figures, single seed descent, pedigree method, and modified pedigree method resulted in 90% of cumulative genetic gain in the fewest amount of cycles. Mass selection and bulk breeding took longer, and we can see that this is especially true with low heritability traits such as seed yield. So this figure is showing the allele fixation rate for the different traits. And we can see that single seed descent, pedigree method, and the modified pedigree method led to the greatest percentage of fixed genes. In the case of white mold resistance, we see that bulk breeding also led to the same amount of fixed alleles. And across all the traits, mass selection had the poorest performance. Onto the Hamming distance. So the Hamming distance is essentially the distance to an optimal genotype. So the lower the Hamming distance, the better. We can see here that mass selection has the highest Hamming distance across the different traits. And we can also see that, interestingly, in days to flowering, the pedigree method did not perform as well as the modified pedigree method, single seed descent, and bulk breeding. Um, so for the average genetic gain per cycle, I showed the different methods, conventional speed breeding and genomic selection. Just as a note for speed breeding, I've omitted the pedigree method and modified pedigree method because speed breeding cannot be used for strategies that have early generation selection. And we can see that for um, days to flowering, speed breeding and conventional breeding lead to similar average genetic gains per cycle, um, but both of these are higher than genomic selection. For white mold and seed yield, we see that there is a clear advantage in using single seed descent in speed breeding over the conventional breeding methods. Next, I wanted to look at the average genetic variance for the different strategies. Um, and I wanted to compare conventional and genomic selection. And the reason is when you have a higher genetic variance, you have more potential to increase genetic gain. However, I did not find any significant differences between conventional and genomic selection for all the different traits. So to sum up the results, the differences between the breeding strategies were found to be statistically significant. The pedigree method, single seed descent, and modified pedigree methods led to the greatest genetic gains in conventional breeding. For low to moderate heritability, speed breeding using single seed descent outperformed conventional breeding that used early generation selection strategies. And genetic variance between conventional and genomic selection for the five strategies were not significantly different. So, in conclusion, dry beans are highly nutritious and sustainable to grow. Plant breeding can be used to increase the yield of dry beans, and computer simulations can assist plant breeders in decision making. Single seed descent, pedigree method, and modified pedigree methods had the best performance. Speed breeding with single seed descent is advantageous, especially with low heritability traits, and genomic selection as a tool to enhance genetic variance is not advantageous over conventional breeding in dry beans. Future studies should consider looking at using genomic selection to identify superior crosses in simulation studies, and to look at incorporating economic decision support tools into QGene to help with resource allocation. I'd like to thank my supervisor, Dr. Valeria Oyas Viegas, Dr. Vivi Arief from Queensland University, my committee member, Dr. Philippe Seguin, as well as my various lab members. Thank you very much. Let me know if you have any questions.